All right, hi everyone. Welcome to Machine Learning for Biochemical Applications. This is a lecture series put together by the BioML Society here at UT. Today we will be going over just the course introduction and then an overview on machine learning. My name is Clayton Kosonocky. I am a second-year PhD student in Andy Ellington's lab, and I'm doing research in using machine learning for chemical and drug discovery. So as I mentioned, this course is a student-developed course by members, graduate student members of the BioML Society here at UT. This is a student-run organization that focuses on performing research at the intersection of machine learning, biology, and chemistry. If you're curious about learning more, we have meetings every Tuesday at 11 a.m. In this room. So definitely check out our website if you want to learn more. So the timing of this course is going to be on Wednesdays from 12 to 1 p.m. It's going to run for 8 weeks. The class format is going to be a 30-minute lecture followed by a 30-minute kind of open Q&A section when you can learn about various other topics if you're curious about like very specific questions. And so the lectures, but not the Q&A sections, will be recorded and posted to YouTube and also the BioML website if you cannot make it. So here's the syllabus that we outlined that we want to cover during this 8-lecture course. The first half covers the fundamentals of machine learning that are relevant towards biochemical prediction tasks. And then the second half is on specific applications and models. So today's lecture, we will cover an overview of machine learning. The next lecture will cover integration with various modern AI tools to help speed up research and coding. The third class will cover the principles of regression and classification tasks, and also their implementation in scikit-learn and Python. The fourth class will cover what are called embeddings, which are the learned representations from these machine learning models, and how we can also utilize this concept called transfer learning to take some of the most powerful machine learning models out there and then adapt them for our specific tasks. Once we get through the fundamentals, we'll go into the application side of things, which we will cover 4 main kind of diverse applications in biology and chemistry. The first one is with AlphaFold, which I'm sure most of you are familiar with at this point. And we'll do a deep dive on that and its ability to help solve the protein structure prediction problem. The second application is in protein design, where we'll cover this one paper on from the Baker Lab on using these state-of-the-art diffusion models to generate novel proteins. The third application is investigating DNA language models for variant mutation effects. And then the fourth is chemical language models for drug discovery. So through all this, we hope to instill 4 main takeaways from this course. The first is to instill a high-level understanding of machine learning so that when you come across these terms in papers and in conferences, you have some idea of what they're talking about, even if you don't know everything about it. And you can get some pointers to some of our slides as well if you want to kind of trace back to these. The second is to help you all understand the principles and use of popular AI tools for biology, chemistry, and programming, ranging from AlphaFold to GitHub Copilot to ChatGPT. The third is to teach you all how to learn how to extend powerful models, powerful pre-trained models for low data biochemical prediction tasks. And I think this one's actually quite important that people won't kind of expect to learn in this course. But a lot of us perform in the biological sciences, we perform these kind of low data experiments that are very high quality data. And a lot of this data traditionally wasn't really used well for modeling because it was too low of a sample size. But we can actually make use of some of the foundational knowledge learned from these large models to actually perform some valuable predictions off of this low data regime. And the fourth is to help inspire some interdisciplinary collaborations between the computational students here and the biological wet lab students. And so without further ado, here's going to be the high-level overview of machine learning. So we are in the age of machine learning. Whether you've seen the self-driving cars driving around West Campus, or you've used ChatGPT, or seen some of these crazy pictures generated from these image generation AI. I don't think I have to stress how important machine learning is becoming, since you're all here in this course right now. So you may be wondering, though, if machine learning is useful for biology and chemistry. So here I have outlined 3 main applications. I kind of lead the answer towards yes, it is quite useful, and it can be even more useful if we kind of investigate some further problems. But the first example here is in protein structure prediction with AlphaFold. This is a protein generated through AlphaFold. And yeah, it's kind of like old news at this point if you're kind of familiar with the field. So I'll move on to some of the more interesting applications they might not be familiar with. But The second one I have up here is protein design with RF diffusion from the Baker Lab. This paper came out a few weeks ago, and it can outline the design of a protein that can fit around a specified constraint. And so in this case, they have this alpha helix that they specified that they want in their protein, and they designed another protein using machine learning to essentially wrap the structure around that alpha helix and include it in the structure. And then they experimentally validated this novel protein to actually fold into that correct shape. And so the third application is we can actually use these models, specifically large protein language models, to better increase our understanding of protein space. So these models are trained on just large, high numbers of millions and millions of sequences of protein data. And through that process, they can learn the evolutionary relationships and structural relationships that emerge from seeing this many sequences. And we can then compare the proteins with one another in this high dimensional space. And so now I want to talk about, I want to go into the ultra-fast primer on machine learning that we're going to cover today. And so in this, we'll cover what is machine learning and how it's different from AI and deep learning, the core concepts and principles of such, a survey of the popular architectures, and the data types that we commonly see in biology and chemistry that we can perform model training on. And then also want to touch briefly at the end on the limitations of using these models. And so the goal of all this, I want to preface, is not to instill a deep understanding or a mathematically rigorous derivation of these concepts. I think there are a lot of much better resources out there than we can provide. And so we have linked to these resources at the very end of our presentation. And so the goal here is, once again, to instill your vocabulary and have you all kind of understand these general terms and how they fit in with one another and how they might also fit into your own research. And so without further ado, AI versus machine learning versus deep learning. So artificial intelligence is the broad category of teaching machines to perform human-like tasks, or tasks that require human-like intelligence. Machine learning is a subset of this, which is specifically accomplishing this goal through having computers learn without explicitly being programmed to do so. So in this case, we can basically show computers data and have them on their own figure out the relationships needed to actually transform that data. And deep learning is a subset of machine learning in which we utilize these brain-like architectures called neural networks. So machine learning is quite different from traditional programming. In traditional programming, we are the ones who specify and design how the data will be transformed from X to Y. So we might say, if you see X, then do Y, or if you see these characteristics within X, then transform it into Y. Machine learning, on the other hand, doesn't utilize these logical statements. Instead, it can learn how to implicitly transform the data from X to Y by just seeing enough examples of X and Y. So it can learn those really complicated, hard to express relationships that would be quite challenging to actually write in if-then statements. So now to dive a bit deeper into this. So traditional machine learning is actually a different term from deep learning, as I had mentioned previously. So in traditional machine learning, we can walk through the process of how this works in this pretty simple kind of cartoony example. So here we have an input, which is a picture of a cat. And our whole goal is to predict whether or not this is or is not a cat. So to do so, in traditional machine learning, we will take this cat image and then extract out various features through the process called feature engineering. And so the features that we might extract from this cat could include things like the length of its whiskers, or how many whiskers it has, or the color of its eyes. And we can feed all these features that we think are representative of the cat into a traditional machine learning model here, which will then create what is called a decision boundary, which when we have our certain features, it tells us if it's on this side of this feature space, then it's a cat. If it's on this other side, then it is not a cat. And so to spend a bit more time here, I want to ask the audience, what features could be used to potentially predict cat likeness from images of cats? Luke. Cool. Philip. Like number of paws or like size of paws? Yeah. Klaus? That's a good point. Yeah. That's a good point. Yeah. If we had like number of wheels, we would need to make sure that cats would have 0 wheels in the pictures. And so like that'd be a key differentiation. But like the task between like cats versus dogs is going to be a much more challenging task that could be harder to actually explicitly specify the various features that we need. So during this process, I listed off some various features that seem to be quite specific towards cats, even though these could also technically be used to describe similar animals like dogs. Um, so yeah, so we have like eye shape, head shape, the proportional sizes between the different body parts, and so on. And so the whole point of this section was to basically get you thinking about what these features and kind of what these features from individual objects that we want to train on could be, because these are going to vary significantly if we're talking about proteins versus chemicals, which are much more complicated to describe than cats. And so deep learning is actually the subset of machine learning, and we don't have to specify the actual features in the process of doing our prediction task in this case. So for deep learning, we can just take the image of the cats versus what we're trying to compare against, and then we can just condense or consolidate this input image into, say, like a one-dimensional vector that represents all those RGB values, and then we can pass them through this neural network architecture right here. And then from that, we can perform the feature learning automatically in the process of task completion. And then it can learn the features and do the classification all within this neural network architecture to then predict cat versus not cat. Are there questions about this slide right here? Or should I progress? Cool. Okay, so to dive deeper into neural networks now. So this is our example that we'll be kind of thinking about, which is our raw image of our cat into this neural network, which will then produce an output. But I think it could be kind of simpler if we still think about this in terms of extracting out the features instead of just passing in the whole image of the cat into this. So we can think about each of these yellow dots as indicating the variable that we want to focus on. So this top variable might be like the proportion of the paws to the front paws to the hind paws. The second one might be like the length of the limbs and so on. The third one could be, you know, whisker length or number of whiskers. And so we can take these input variables and we pass them into these circles here, which I'll get in depth on what these mean. These nodes are called neurons, and these are the base computational unit of neural networks. And so the whole architecture of neural networks basically follows an input into these concurrent hidden layers that kind of just like pass one another. And these are basically just stacks of these neuron computational units, which then can feed forward into the next layer and then into the final layer to then do our predictions. And so I'll go more in depth on what this means now. So to understand this more, we have to first go in depth on what the neuron actually is. And so this, once again, is the computational unit of neural networks. And so the neuron is actually quite simple. This is basically just a weighted sum of the inputs going into this little, into the neuron. And then it is that weighted sum plus, so x1, x2, x3, these are our input variables. And each one of those will be multiplied by their respective weight values, W1, W2, and W3, to create the weighted sum Z, which is equal to that sum plus a random bias, or not a random, a learned bias term. And once we have this weighted sum Z, we can pass it through what is called an activation function. And so these activation functions are what are used to create nonlinearity in our function approximation. And so we need this nonlinearity so that we can approximate more complex functions, and without which we would only be able to approximate a smaller subset of functions. And we would actually be able to, if we didn't have this nonlinearity, we would not even need this whole neural net architecture, as we could represent our whole neuron in terms of a single linear system, which is not what we're trying to do. So this activation function, this one's called ReLU. This stands for the rectified linear unit. It is a fairly common activation function. This one just takes the input going into it. So in this case, the Z value, which is the weighted sum of the inputs. And then if that value is positive, it'll just return the positive value. And if it's negative, then it'll return nothing instead. And so this then creates like various kind of branches of, if an input goes in, it'll have like zeros coming out sometimes versus positive values coming out. And it can create this sort of like learned network of essentially an approximation of these conditional statements. And so you might be wondering at this point how these weight values are actually calculated, and also this bias term that I mentioned. And so basically, the whole point of neural network training is to have the model kind of converge automatically to learn how to best weight the input data to then accomplish the task at hand. So to do this, we need to first like give the model a random guess as to what we think the weights would be for this weighted sum. And so the first step is to initialize the weights randomly. There are more complicated ways to initialize the weights that sometimes lead to better success in prediction tasks, but I'll not be going over that in this lecture. I just want to mention as well, if you have any questions, please raise your hand during this lecture and I can answer them on the fly. So once we initialize our weights, we can then pass in our sample into our neural network. And I also want to stress briefly that this training process I'm illustrating in context of a single neuron, but in reality, this is dealing with all these interconnected neuron systems. But this should serve pretty well to give you guys a high understanding of how this works. So yeah, we take our sample and then we input it into our neural network. And we can basically, the input sample goes into these X values, gets multiplied by the random initialized weights to get this weighted sum. And then it passes through the activation function to then produce the output. And so if we were doing some sort of classification task like cat versus not cat, this output would probably range from 0 to 1, where 0 is not cat and then 1 is cat. And if we have our initialized weights and we showed a picture of a cat, it is pretty unlikely for it to actually get the correct value. And this is because it doesn't yet know how to properly weight the input data to then get the actual correct estimation of whether it is or is not a cat. And so to help steer it towards being able to predict better, we need to first see how wrong it actually is on a given guess. And we do this by computing what is called the loss function. So the loss function is just the difference between the predicted output of the neural network to the true output. And the specific loss function that you use depends on the task at hand, but you can think of it as just a simple difference between these two values. And so in our case, if we predicted wrong, then our loss function is going to be quite high because the difference between our true value and our guess value is going to be not 0. It's going to have some significant value to it. And so to help calculate how we should adjust these weights with response to this loss, we need to compute what is called the gradient of each of the weights with respect to the loss. And so if you've taken calculus, you might be familiar with this. But essentially what is happening here is each of these weights can be represented as a function, or sorry, the loss can be represented as a function of each of these weights. And so therefore we can take the gradient on each of these weights, which is essentially the, let me back up. So we can represent the loss function as a function of all these weights, W, and then we can get this space, which is as we change the weights, the loss function will either go up or down. And we want to basically optimize the loss function to go down as we change our weights so that we can minimize the loss and make our predictions more correct. And so to do this, we take the gradient, which is essentially just calculating which direction we need to move for each weight to then make a lower loss. So once we have that gradient calculated, then we can do what is called backpropagation, which is essentially take our loss and then shift all of our weights in a direction that can then help lower that loss. And I have posted a video here that goes a lot more in depth on this, which I recommend checking out if you're more curious. And so essentially, once we have that for one sample, we really want to just do this for batches of samples so that we can get a more accurate estimation of what the true gradient is over the task that we're looking at. And so we can train our model in batches of data and then adjust the weights after each batch to then make the loss get lower and lower and lower for each batch of data, which then would cause our model to then converge and get more accurate at the task at hand that it's being trained on. Are there questions about this section right here? There's a lot of methods to prevent that, and we're going to go more in depth on that in the third lecture. But essentially, we can just take some data that we hold out from the model, and we can evaluate it on that data instead. And if it seems to perform very well in the training data, but poorer and poorer and poorer on that other data, then we know that it's overfit. Yes. I think it depends on the task. Yes. Yeah, it really depends on the task that you're trying to do, which is unfortunately the question, the answer that I'll give to a lot of these questions. But to go maybe help out a bit more, there's advantages to doing both. If you specify the actual features, then you're alleviating some of the burden that the model has to learn, because it won't then have to learn, like if you're just feeding it images of cats, it has to first understand like what paws are on its own, or these like basic objects and everything. And so that's, if you just show the model like pure pictures of cats and hope that it can learn these things, then it would take more computation to probably do. but it also, on the pro side, it might capture relationships that we can't really specify. And so there are some domains where people feed in just the raw image, and then they can also supplement them with some specified features if they don't think that the model would capture it. Yes. Are you saying like the loss function as a function of all the weights, like the whole space? Yeah, I do not think we know the shape of this curve, and we just do it iteratively through our process. I believe. I will look into this after. I'm pretty sure that's the case. Yeah. Okay, so the next section that I want to talk about is the various data types that we encounter in biology and chemistry, and then the neural network architectures that we find commonly associated with these in the literature. And so the first data type is vectors, and so I want to first talk about one-dimensional vectors, which are essentially just like 1 by n vectors. So here we have a 1 by 4 vector of 4 different variables, and we can pass this into what we've been looking at this entire time, which is a feed-forward neural network. And so this vector of 4 variables goes to this hidden layer of as many neurons as we want. In this case, they're showing 3 neurons. And so this variable 1 goes to neuron 1 and hidden layer 1, and also neuron 2 and neuron 3, and so on. So this is essentially fully connected in this case. So each neuron views every single part of the input. And the reason why we have multiple neurons within this hidden layer is to allow some neurons to capture different functional relationships, which can then be pieced together in the final output aggregation step right there. And so these are very good ways to do regression and classification tasks, which we will talk about in lecture 3. So commonly in biology and chemistry, we deal with coordinate systems and structures. And so here I have shown arbitrary coordinates and for proteins and chemicals right here. You can find these in PDB files for proteins or in like SDF files for chemicals. And essentially, it's just a 3D representation of these objects. And we can deal with these either, one naive way to deal with these is to just smash down that 3D coordinate system into a one-dimensional vector arbitrarily and then pass it into a feed-forward neural network. That has the downside of losing a lot of the relational information between these coordinates. And we can actually preserve this local relationships that the local relationships that we see with these mathematical operations called convolutions. And so these operations, with using these operations, we can take, say, an image in this case, and then we can reduce the dimensionality of this image until it's just a one-dimensional vector, in which case we can then pass it into a feed-forward neural network to do a classification or regression task. So these convolutional neural networks, you can think of them as ways to basically encode spatial data into vectors that we can then perform further tasks on. So if we wanted to instead generate spatial data rather than encoding it and doing tasks on it, we can turn to some generative models. And in particular, I'm outlining a new model, or a fairly new model, a state-of-the-art one called diffusion models, which are used to generate these hyper-realistic images shown in the bottom right. And so these models have recently been adapted towards biochemical tasks like protein design and molecular design. And I showed at the start an example of that for proteins right here. And we'll be going over this in more depth in lecture 6 when we cover RF diffusion. Whoops, cool. So the next data type are called graphs. And in this context, a graph refers to the connectivity between nodes and edges in a node and edge-based system. Sorry, that did not sound right. The graph is, yeah, it's this array of essentially nodes and their connectivity represented as edges. And so in this case, we have this molecule right here, and we can think of each atom as a node in the system. So we have 4 Cs, and we have this many hydrogens right here. And then basically what we have, we can encode the connection between these with various values based on the number of bonds between them. So zeros would represent no bonds between these atoms, ones represent one bond, and twos represent double bonds between them. And from that, we can create this graph graph-based visualization or this graph-based representation of our molecules. And so you might be wondering why we even bother doing this. And the advantage to graphs compared to structural coordinates is that they're rotationally invariant. So it doesn't matter which direction we feed these proteins into our model we can just feed them just only the pure connectivity information. And so, whoops, this is acting funny. Cool. So we can feed these graph representations, like from this molecule right here, into a neural network as well. And this is using graph neural networks. And specifically, we're looking at a graph convolutional neural network, which kind of combines some of the previous convolutional neural network design that I showed earlier. And so here we have this graph system, and we can reduce this dimensionality to the point where we can once again throw a feed-forward neural network on top of that and then do our regression or classification task. In this case, we're predicting the drug side effects given a graph representation of a molecule. And so we'll be covering this a bit more in depth in lecture 5 when we go over AlphaFold, which utilizes graphs in its neural network architecture. So the next data type are strings, and these are essentially just sequences of characters or numbers or punctuation. It doesn't matter. And so here I have shown an amino acid string, which is you can encode most proteins into a string of amino acids, which I'm sure most of you are pretty familiar with. Here is a table showing the conversion of that. It's pretty standard. You might be less familiar with a different representation, which is for chemical strings. So you can actually convert any arbitrary chemical into a one-dimensional string representation using some systems. Specifically, this one's showing the simplified molecular line entry system, or SMILES system. And so we can go from this arbitrary chemical structure to a string, and then we can go back into that same structure in the decoding process. And so strings represent an interesting different representation compared to protein coordinates because it's a much more dense data format. But you might be wondering how we actually feed these into our models, considering our models are doing mathematics on the input data, and strings are not inherently numerical values. So we accomplish this through the process of tokenization, which is essentially how these models can learn to understand strings. And so this case is showing the tokenization of a SMILES string representing a molecule right here. And what we do in this process is we break down the string representing the molecule into a bunch of, or a sequence of substrings here. And then with these substrings, we can then represent or assign each of these an integer value, so that every time this token or this substring comes up, it has that same integer value associated with it. And then here, the model will then take in this vector, which is called, which I'm calling the token vector right here. And so this is now an integer sequence representation of the input sequence. And so these sequence or even time series data, or primarily in the context of string data, can be learned from various models, but I'm going to go over 2 of them briefly. The first one are called recurrent neural networks. And these have recently been sort of outperformed by this newer model, which is the state of the art in this field, called transformers. And so transformers use this concept called self-attention, which we will go over more in depth in lectures 4, 7, and 8. But these models are what ChatGPT is based off of in all these large language models. And so they're quite powerful systems. And so this is essentially the summary slide of the architectures and data types. We covered quite a few architectures that are commonly seen in the literature, but this is not a comprehensive view of these. And there are definitely some kind of like nuances to how these are actually applied. We hope that this serves as a nice overview and kind of reference point to think about these models and these data types. And so with that, I want to leave you with one kind of final slide on the limitations of neural networks in general in these models. And that is that the inference scope is limited by the training data. So what this means is that if you trained a model on only 2 classes of data, like only cats or dogs, then you could definitely distinguish between cats and dogs and perform inference on similar samples of cats and dogs. But if you were to all of a sudden take your model that was trained on cats and dogs and then start to inference it on robots and try to make guesses on these robots, robot images or whatever, then it would definitely it should perform very poorly, because those robot images would be out of distribution compared to your training sample. So it's really important to understand what data your model was trained on so that you can know the applicability of that model and its limitations and what you can use it for or not. And so today I covered a fairly high level overview of machine learning to get you all up to speed on some of the core concepts. Here are a bunch of great resources I would recommend checking out if you want to dive deeper, either still in the high level or if you want to go more into the mathematics behind how these work. And so that is all I have for the lecture today. And I guess we can do a Q&A section. Um, we can start with the whole, uh, I'll just answer questions up here for the Q&A, and then we can break and do like individual questions if you want. Thank you. Yeah, so next class we're going to be, oh, yes, cool. The question was, are we going to have a chance to practice on the concepts that we learn in this class? And I think uh, I believe we will make some resources available to do this. Specifically, mainly in the application side, we'll be providing links to use these applications online so that you can play around with them and get a feel of how they actually work. And for the programming side of things, in the next class, we're going to be getting you all up to speed on kind of like getting situated with setting up your coding environments if you want to start doing some of the programming. And we will also help you get integrated with some of the modern tools like these like auto completion tools for coding, which can help things out and speed up your life in programming for biological tasks. And for the regression and classification class, I believe we will be providing some simple code to get you kind of able to play around with these concepts as well in these models. Sure. Hmm. It's all partial derivatives. Yeah. And so a lot of the partial derivatives are actually a function of the previous magnitude of the weight going into it. And so they all kind of can vary by different amounts. And so they're not just like all shifting by the same amount every time. Yeah. Did that answer your question? Cool. Yes. It really depends on the task that you're doing. And it also depends on how optimized these models are. Some of these large language models typically require like server-side computation, but people have been taking these models and optimizing them as much as possible to the point where they can run fairly fast on like basic laptops like MacBook Airs and everything. So it really just depends on the model that you're working with and the data types that you're working with, and so on. Yeah. It, yeah. Yeah. Hmm. Hmm. The architecture of the neurons. Yeah, that's a good question. So the question is, can we, is the number of neurons, or say like the number of layers, even I would extend that question, is that like preset, or do we learn that during the process? And so that is typically defined through this process called cross-validation, where, or let me back up. So the number of neurons and the number of layers are called hyperparameters because they kind of extend over the whole model. And so they're not learned given a single model training process, but you can figure out the optimal orientation by training several different models with several different numbers of neurons or numbers of layers and so on. And then you can do cross-validation to then figure out how it performs best on your validation set, which is another holdout set that you have that is also separate from the test set. And so we go into that more in the same classification regression task, I believe, on the data splitting. Um, but yeah, so it's not, there are some heuristics on kind of guessing your amount that you should start with and what you should kind of vary with. But it really does depend case by case. All good. Yeah, so that's a good question. So I think they were inspired initially from just like Gaussian diffusion processes, and I'll go into this much more in depth during that class. But how they actually like look when they're doing their inference sort of looks like diffusion because it's all this like kind of like conditional transitions from like each step to each step to each step that sort of mimics the diffusion process somewhat. Um, yeah, but we'll go over that more in depth. It is. Okay, cool. It works for some reason. Mm. Yeah, that's a great explanation. Any further questions? Yes. Sure. Yeah, so I don't think we would give the estimate of the weights as an input, but we could initialize our weights differently. And so that's kind of similar to the concept of transfer learning, which we'll talk about later. And so in transfer learning, we can take these models that were trained on one task, and then we can adapt them for a separate task, which is essentially serving as this initial guess, this really well-educated initial guess of how to perform the task. And then the model can learn from those weights to actually adapt it for whichever task it may need. So using that, you can reduce the number of data that you need in your task like significantly and converge on your task way faster and typically with higher success. Did that answer your question? Cool. Yes, Luke. I would call them more data engineering personally, but I think it really depends on the person. Yeah, yeah, I think so. Hmm. Hmm. I think that's a tricky question to answer broadly, and I don't know if I'm completely skilled to answer it representatively. But I do know that if you did train, say, like a model just to do a simple computation with like 2 goes to 4, 5 goes to 7, then it should, I believe, be able to encapsulate that transition. Um, there are downsides to using single neuron systems, and they can't really approximate some higher order functions, like the not or sorry exclusive or function. A single neuron cannot represent that. But as you add more neurons, then it can actually start to represent these functions that are easier to program, but harder to actually have a single neuron perform. Um, but the whole task of explainability is sort of what you're getting at a little bit, which is like, can we pull out what it's actually doing? And that remains still basically an outstanding challenge in the field as to how to like best explain various model types. And there's different ways that you can try to kind of do it based off the specific model. Like for convolutional neural networks, which are focusing in on like images, we can have it highlight the parts of the image that are most relevant to the analysis, which I could throw in some slides at some point relating to that. And similarly for transformers, which work by this attention mechanism, we can do some sort of analysis, some proxy of analysis on what it's paying attention to in the input to make its decision. Um, yeah, did that answer? Cool, awesome. Great. We can braid for today. Come see me afterwards if you have any more questions. Thank you.